Okay, so this is part 2 of our chapter 10 lecture. And in the beginning of our lecture, we talked a little bit about antibiotics, antimicrobials, some of the terms that we'll be using throughout the class. And then we went through the first two types of mechanisms of action, one being antibiotics or antimicrobials that target the cell wall, and the second being the cell membrane. Now let's talk about the inhibition of nucleic acid synthesis. And we have a number of different antibiotics that inhibit nucleic acid synthesis, but we're going to focus in on quinolones. And so you can see here. Um, okay, so essentially, DNA replication, nucleic acid synthesis could be DNA replication or it could be mRNA transcription, right? So in terms of any pathogen that is growing inside of a host, it's absolutely going to need to replicate its DNA as it grows and reproduces. And so this is going to be absolutely essential to that pathogen continuing to grow and replicate or reproduce within a host. So when you have drugs that actually target replication, this can inhibit or reduce the capacity of that pathogen. Particularly, quinolones actually inhibit the activity of DNA gyrase. And DNA gyrase, if you remember back to chapter 7, is one of our topoisomerases, which is actually in preventing supercoiling of the DNA as it's being replicated. Now, there are some DNA gyrase-like molecules that are also working within transcription. And so, in fact, quinolones inhibit the replication of DNA. So here's your DNA gyrase. If you inhibit the DNA gyrase with a quinolone, then it actually prevents that pathogen from continuing to replicate its DNA and reproduce. But also it can inhibit transcription, and so the production of mRNA off of DNA templates. And so quinolone actually inhibits both processes, so the pathogen is not able to produce DNA or mRNA. Okay. We also have an antibiotic called rifampin, and rifampin inhibits the RNA polymerase, so specifically inhibiting the production of mRNA without inhibiting the production of DNA. But it actually binds to the RNA polymerase, which is copying or making a copy of the DNA into mRNA, and it prevents mRNA production. So rifampin is also an antibiotic that inhibits nucleic acid production. Then you have some nucleic acid inhibitors. Some of these particularly are important to antivirals or important antivirals. And particularly there's these nucleotide or nucleoside analogs, which essentially they mimic nucleotides, but then they're made and they're not functional. And so when they're used to make DNA or make RNA, then that DNA or RNA is rendered non-functional. And in that way, the pathogen is inhibited or the virus is inhibited. That is particularly useful against viruses. And actually, the first of these nucleotide and nucleoside analogs was the first effective drug against HIV, which was made by or was developed by Gertrude Elion, who received a Nobel Prize for her research in 1988. And so there's also an optional video on Gertrude in the description for this class that you're welcome to watch, but it gives you some information on how she developed some of that research or a number of different techniques and a number of different kind of diseases that she researched. Okay, and so here's just an image of these nucleoside or nucleotide analogs, which is essentially non-functional nucleotides that take the place of real nucleotides and in that way render the pathogen, either slow it down or actually inhibit it overall. Okay. Okay, so those are some examples of how some of our antibiotics are inhibiting the production of nucleic acids like DNA and RNA. Okay, Now let's move on in the step. So we talked about DNA, we talked about RNA. The next step in our central dogma is proteins. And so you do have some antibiotics that specifically inhibit the synthesis of proteins, but then you also have some antibiotics that inhibit enzymes within metabolic processes. So first we'll talk about inhibiting the synthesis of proteins, and then we'll talk about metabolic pathways and inhibiting metabolic pathways. Okay, so important to the discussion, if we're talking about inhibiting the synthesis of proteins, we have to remind ourselves that prokaryotic ribosomes have a slightly different structure and weight than eukaryotic ribosomes. And it is in this way that we can have selective toxicity of chemicals that inhibit protein production, specifically in prokaryotes. But then also remember that antimicrobials will not work against against viruses. And so we do have to have a different type of inhibition when we're talking about virus protein synthesis. Okay, so what are some of the mechanisms of action for inhibiting protein synthesis? Remember that we have two subunits. We have a larger subunit and a smaller subunit. So in a prokaryote, that's going to be 50S and 30S. And then you have your mRNA, which moves through that. And so you have a number of different antibiotics, including macrolides, that will actually bind to the 50S, which is the larger subunit, and prevent the peptide chain that's forming out of that larger subunit from continuing to be built. And in that way, it stops protein synthesis. Then you also have some antibiotics like tetracycline that bind instead to the 30S subunit, which is the smaller subunit, and just block the movement of tRNAs, and in that way inhibit protein synthesis. And then you have your aminoglycosides, including streptomycin, which also binds to the 30S subunit, but it actually impairs the proofreading and the proofreading of the mRNA. And it results in proteins that are not functional. And actually that can be really detrimental to the cell. So this tends to be specifically bactericidal, whereas some of these other ones like macrolides and tetracyclines are more bacteriostatic. So they stop the bacteria's ability to do its various functions without necessarily killing it. But these faulty proteins actually make this more likely to be bactericidal, the streptomycin. Okay. And so this image just goes through some of those different actions as well. Then in terms of inhibiting protein synthesis, particularly in viruses, well, we have these antisense nucleic acid antibiotics or antivirals that essentially they bind to the viral mRNA and in that way prevent protein synthesis. So since that literally the antiviral is an antisense mRNA, and that mRNA binds to the sense mRNA, and in that way just can't bind to the ribosome, and then no proteins are formed. And then you also have this example where you have some antimicrobials that will actually bind to the start codon on the mRNA. And in that way, the whole complex never forms and the mRNA is never transcribed. And so that is some antimicrobials against gram-positive organisms. And, more generally, bacteria can also work in that way. Okay. And so in that way, we can use different antimicrobials to inhibit protein synthesis. And oftentimes that's bacteriostatic because that organism is no longer able to make the proteins that it needs for its functions, but it's not like physically broken apart. We said there are some exceptions to that, particularly when we're looking at the streptomycin, which actually results in faulty proteins, and that can be really detrimental to the cell. Okay. There are some side effects though, because we also, as the host, have ribosomes, even though they're still structurally different from prokaryotes, but sometimes there can be side effects on the liver and the bone marrow to using these antimicrobials that inhibit protein synthesis. Okay. So we've talked about antimicrobials that inhibit the cell wall, particularly the synthesis of the cell wall, the cell membrane. We've talked about, as well as the cell membrane, we've talked about antimicrobials that inhibit production of DNA and RNA. We've talked about antimicrobials that inhibit protein synthesis. Now, let's talk about antimicrobials that inhibit specific enzymes or proteins within metabolic processes within the cell. Now, when we talk about metabolic processes, we also always want to go back to that selective toxicity, and we definitely need to have antimicrobials that are targeting metabolic processes that are unique to pathogens rather than to the host. And so oftentimes we look for metabolic pathways that are present within the pathogen rather than in the host. One example is the group of antimicrobials called sulfonamides. And essentially what they do is they target the folic acid pathway. And so folic acid is a vitamin that is needed across the board for all living organisms. But prokaryotes tend to make it on their own. They actually synthesize it, whereas humans or animals, we actually have to ingest it. We can't make it on our own. So we don't have the metabolic pathway to make it. And therefore, if we target with an antimicrobial or an antibiotic that chemical pathway, then it's selectively toxic to the pathogen rather than the host. And so the way that sulfonamides work is there are a couple of different sulfonamides that work in different places within this metabolic pathway. But many sulfonamides, what they do is they're actually competitive inhibitors of the beginning of this process. So we have this PABA molecule, which is part of the beginning process of folic acid production. That's a para-aminobenzoic acid, PABA. And you need that in the beginning enzymes within the pathway so that you can eventually make folic acid. But what sulfonamides do is they mimic PABA, and so they're competitive inhibitors of that site on the enzyme. And so if the sulfonamides bind instead, then the PABA cannot bind. Right here, we have an image here. And in that way, folic acid is not produced in those pathogens. And in that way, their activity can be first, mitigated, and then eventually they can that antimicrobial will address that infection. Okay. And so there are also some antivirals that can target various metabolic activity or proteins within viruses. And there are also some other classes of antimicrobials that inhibit various parts of the folic acid pathway. Okay. Okay. So that is all of our different mechanisms of actions for prokaryotic pathogens. So you see here all of our different mechanisms of actions if we're talking about a living prokaryotic pathogen. But we want to just have a final discussion about antivirals. So viruses being separate, non-living disease-causing agents. And remember, we said that antibacterials or antimicrobials do not work against viruses because viruses don't have all of these different metabolic activities and the production of DNA and RNA and proteins all in the same organism and so on and so forth. And so this is actually one of the issues with kind of medical prescription of antibiotics is oftentimes antibiotics are prescribed for a viral infection and then they're not effective against that disease, but they are effective in potentially, eliminating part of the microbiome of that person, decreasing their ability to kind of, their healthy overall holistic ability to fight off pathogens, and then also increasing antibiotic resistant strains of bacteria because of the overuse of antibacterials against viral pathogens, which do not respond. And so just being thoughtful of using antivirals instead of antimicrobials when it's a viral infection is really important. We did say that the most basic components of a virus is that they should have a nucleic acid, so genetic information, and a capsid, so that nucleocapsid, and sometimes they also have an envelope. And we said that it is most important, it is essential that that virus can attach to the host and it can release its genetic information into the host. And so you have attachment and you have that release of genetic information. And so we have just two antivirals that we'll talk about here, but there are many antivirals. Fluconazole blocks viral attachment, particularly in picornaviruses like cold virus, poliovirus, and some other viruses. And then aldrone prevents viral uncoating within the host. And so you can see here, this is just a reminder that we have our virus attaching to the host and then eventually fusing with the host and entering the host. But if you can block that attachment, with an antiviral, for example, then you can block infection. Whereas araldone is more specific to poliovirus and it prevents uncoating. So if the poliovirus were to enter the cell, but then inside of the cell it remains coated, then it can't release its genetic information and infect the host. Okay. So that is just a survey of all of the different potential, or a number of the different potential mechanisms of action for antimicrobial agents and antivirals, and then just a couple of examples of each. And so this might be a good time to pause the video, review your notes, cross your T's and dot your I's. That will essentially, a summary of that discussion will be our discussion post for D2L. And then you can come back into the video and we'll talk a little bit about how we use antimicrobials and how we overuse antimicrobials, and we'll finish off our discussion with some best practices. Okay, welcome back, folks. So we talked about how antimicrobials can be used and how they're kind of mechanisms of action with that at the molecular or the microbial level. But then when we're talking about widespread dissemination of these antimicrobials, we can think back to that example of penicillin and Fleming, where we have the discovery of penicillin, but it's actually not widely available until decades later. So the ideal antimicrobial agents tend to have all of these characteristics that you see here: readily available, inexpensive and accessible, chemically stable, easily administered, non-toxic and non-allergenic. So that gets back to selective toxicity, which we talked about, and also selectively toxic to the pathogen, and specifically the pathogen and not the microbiome, which is not necessarily stressed in your book, but that's really important because having a healthy microbiome, as we already know from our previous discussions, is an important part of having a strong immune system. And so you have some tables in your book that talk about the different antibiotics and whether they're narrow or broad spectrum and kind of who they tend to target. So I would recommend looking over those, and you can see some of these like tetracycline, a little bit more broad in its spectrum, but then polymyxin tends to be more specific, narrow or limited in its spectrum. And you can kind of look at those tables there and get a little bit of an idea. In terms of effectiveness, you really want to test a specific strain against a set of antibiotics so you can see if those antibiotics are effective against that strain. And that is because of horizontal gene transfer, which we've talked about in chapter 7, and we've also been introduced to in the lab. where essentially microorganisms can actually be the same species, but they can actually transfer antibiotic resistance genes. And so you want to know if that specific organism is actually susceptible to the antibiotics that you're going to be administering in the clinical setting. We will actually do this very specific susceptibility test in lab, where we'll take our strains within the lab and expose them to different antibiotics and see whether or not they respond. So if you get a zone of inhibition on a lawn where you've added these little paper discs that have antibiotics infused in them, then that means that those organisms, those colony forming units around the disc of antibiotic, the antibiotic discs, they were actually killed off and they were actually inhibited by that antibiotic. Whereas if you don't see a zone of inhibition, like in this one here, then that means that that organism does not respond to that antibiotic. And so in a clinical setting, if you have an infection in a patient, you actually want to do a test like this where you can actually see, okay, these antibiotics that I expect to be effective against this organism, are they actually effective, or is this organism potentially antibiotic resistant to some of these antibiotics? And you can very, very easily with one of these tests, these susceptibility tests, see that. Sorry, I moved through the slides accidentally. Also, when you're administering an antibiotic, you want to know what's the correct dosage of antibiotics. And so there are a number of tests. So this minimum inhibitory concentration test just allows you to make wells of your specific organism against a concentration of the antibiotic. So this is actually a serial dilution of the antibiotic from, in this case, it looks like least concentrated to most concentrated, right? So here you have your positive control all across this line here. You have your negative control here, and then you have your organism against multiple different antibiotics. So from A to H, these are different antibiotics. Here you have the lowest concentration of that specific antibiotic, and then you just increase the concentration until you see that bacteria no longer grow or respond. You say, okay, that's a concentration that is effective against this organism. You can see that happens earlier in some of these antibiotics. So see, it grows, it grows, it grows, it grows, it grows. Oh, its growth is impacted here, and then there's no growth here. And same here, it grows, it grows, it grows, it grows, and then no growth here. And so that gives you an idea of the concentration that you might actually want to use for that particular antibiotic against that particular strain of organism. You can actually combine these two tests into one with these e-tests, where you have a strip that has different concentrations of the same antibiotic rather than having multiple different antibiotics. And you can see here the concentration is highest on this end of the strip and lowest on this end of the strip. And you see that organisms on this end in this lawn are most impacted by the antibiotic, and then the impact tapers off as you go lower and lower in the concentration that those organisms are exposed to. You can also do a serial dilution in a tube of an antibiotic mixed with a slurry of your specific organism, and you can plate those and see what is the most effective concentration. But it is important to administer the correct amount of antibiotics so that it is effective against the pathogen, but it is not, over-administered. How are these administered? Well, it just depends on the antibiotic and how it's stable both outside and inside of the body. And so you could have a topical administration, which is also called transdermal administration of a drug. So like bacitracin is also oftentimes administered on the surface of skin. But then you could have oral administration, intramuscular administration, or intravenous administration. So penicillin, for example, depending on the patient and their situation, might be administered in different ways. And so depending on your antibiotic, it could be administered differently to the patient. Okay. There are sometimes side effects, though, right, to antibiotics. And so when you're considering how you're going to administer an antibiotic, you're going to consider the strain that is the cause of the infection, what antibiotics it's susceptible to, but also the characteristics of that patient. So are they pregnant? Are they immunocompromised? Are they a child? Are they allergic to different types of stabilizers within these antibiotics, so on and so forth? And then you're going to administer the correct drug based on all of those different factors. Okay. Let's see. We're coming to the end here. Oh, that's just a little fun art project. Antibiotic resistance, though, is the last thing that we want to talk about because, as we know, organisms evolve over time so that they can evolve to be antibiotic resistant over time. So, beta-lactams are a great example. They're organisms that have the enzyme for something called beta-lactamase, which is an enzyme, ACE, that targets beta-lactam rings. So we said there's that ring structure in beta-lactam antibiotics. And it targets that ring structure and breaks it down, and in that way makes those organisms antibiotic resistant to beta-lactams, right? And there's a lot of different ways that organisms can become resistant to antibiotics, including horizontal gene transfer, which exponentially speeds up the process of antibiotic resistance because organisms that develop antibiotic resistance can share it horizontally within their own generation among their peers. And in that way, we as humans, as a medical system, as a society, have to be really, really thoughtful of how we use antibiotics so that we're not actually promoting antibiotic resistance. And over history, we have not done that. We have indeed overused antibiotics. And you can see, I mean, the first mass use of antibiotics is just during the Second World War. So these are new tools that we have. And we have overused them. We have used them against viral infections. We have put them into commercial items, antimicrobials and antibiotics, to an extent where we've actually overused them and encouraged antibiotic resistance throughout the environment and throughout communities of bacteria. And this is a really big problem because we will continue to develop antibiotics and we will continue to discover new antibiotics, but our capacity to develop new and find new antibiotics will eventually taper off as we become more and more familiar with what's out there. And so if we are rapidly encouraging antibiotic resistance, but also tapering off in terms of the new antibiotics that we might discover or be able to develop, eventually there'll be a crossing point where antibiotic resistance will supersede our capacity to actually maintain these pathogens and organisms with antimicrobials. And so this is actually a really, sorry, I keep doing that, kind of a really urgent issue within microbiology and medicine generally. So I'm going to show you these two videos, and then we're going to end on a positive note of what are some of the things that we can do to help discourage this overuse of antibiotics and antibiotic resistance. So here we go.
A big challenge to medical science today is the growing problem of antibiotic resistance. A pathogen that is resistant to a particular antibiotic will not be killed or inhibited by that antibiotic. It has gained that resistance either by random mutation or through gene transfer from another bacterium. In a particular population of bacteria, there may be a few cells that are resistant to the effects of a particular antibiotic. When the patient is given a particular antibiotic, that drug will kill or inhibit the growth of all the sensitive bacterial cells. This allows the resistant cells to multiply freely in this less crowded environment. One way that bacteria can acquire resistance genes is through mutation. If a mutation occurs in a sequence of DNA, this could result in a different amino acid sequence. Consequently, this different sequence could produce a new enzyme, perhaps one that can interfere with the action of an antibiotic. Bacteria can also acquire resistance genes by horizontal gene transfer. One type of horizontal transfer is transformation, in which a cell picks up a piece of DNA from its surroundings. Or a bacteriophage can transfer the resistance genes from one bacterial cell to another in the process of transduction. Finally, resistance genes can often be found on small pieces of DNA called R plasmids or R factors. These R factors are often transferred via conjugation between bacterial cells. Bacteria can resist the effects of antibiotics through several mechanisms. They may secrete enzymes that destroy the chemical structure of the antibiotic. For example, penicillin-resistant bacteria produce an enzyme called beta-lactamase. The beta-lactamase enzyme breaks the beta-lactam ring in the molecule of penicillin and related antibiotics, such as cephalosporins. This prevents the antibiotic from disrupting the peptidoglycan cell wall structure. Other bacteria prevent antibiotics such as tetracycline from passing through their cell membranes. They do this by changing the structure or charge of pore proteins. Even if the antibiotic can penetrate the cell, a resistant bacterium can make it impossible or more difficult for the antibiotic to bind to its target. Sulfonamides are antibiotics that compete successfully with PABA for an enzyme binding site in the folic acid synthesis pathway. Resistant bacteria may have evolved an enzyme with greater affinity for PABA, thus making the sulfonamide less effective at blocking the folic acid pathway. Other resistant bacteria have developed a method to obtain molecules they normally synthesize directly from their environment. Therefore, they aren't affected by the blockage of the biosynthesis pathway. Finally, some resistant bacteria have membrane-localized efflux pumps that remove some antibiotics from the cell. Some resistant bacteria have a greater number of efflux pumps, thus increasing their ability to eliminate the antibiotic. In some cases, bacteria have also been able to increase the spectrum of drugs that are eliminated by these pumps by making pumps less specific.
Okay. So you can see some of the ways that bacteria are protecting themselves against antibiotics. And that's the dance of life, right? Is that you have an organism that outcompetes another organism, and then humans say, oh, we can use that chemical to outcompete the pathogens that are trying to take over our bodies. And then those organisms or those pathogens then develop ways to kind of combat those actions. And that's evolution. That's kind of like the dance of life. And so it's really important to consider how we can actually be more thoughtful in our use of antibiotics so that we can reduce antibiotic resistance and or not really reduce antibiotic resistance, but the rate in which we see new strains of antibiotic and multi-antibiotic resistant organisms. Now, I do want to bring us back to our discussion of social and medical injustice because access to antibiotics is another way that we actually see the promotion of this types of injustice. And so um, if antibiotics were very widely accessible and vaccines, we could actually very likely you know, kind of administer them broadly and then see a decrease in antibiotic resistant strains and some of these pathogens. Same for vaccines. You could see a decrease or potential elimination of things like polio, for example. But when we have inequitable access to vaccines and healthcare systems that are functional, then we actually see the promotion of some of these diseases or antibiotic resistance. That book, Everything is Tuberculosis, talks a lot about this because we do have regimens that would allow us to potentially eliminate different types of tuberculosis infections, but because those regimens are not accessible to people or they're not administered in an accessible way, then you continue to have this on-off system where people are exposed to antibiotics but not over a long enough period of time to actually eliminate the tuberculosis, and then you actually increase the likelihood of antibiotic resistant and multiple antibiotic resistant tuberculosis strains, which is really dangerous just globally, right? And that's not because we don't know how to address antibiotic resistant tuberculosis, but it's because we don't, in a just and accessible way, administer those antibiotics and those treatments. So it's really important. So what's the good news is we have some tools, right? And so thinking about how we can slow down this promotion of antibiotic resistant strains, we know we actually have all of these tools and we know all of these things. It's really about applying them. It's really about applying them in a system that is just and it's not focused on capitalistic gain, and instead it's focused on access to healthcare broadly for, people globally and across the world. So what are some of the things that we can do? Use antibiotics when necessary. So in a clinical setting, that means use antibiotics, the appropriate antibiotics against the appropriate infections or diseases, and do not use them in the case that that infection is a viral infection. Instead, use antivirals for that. Maintain the correct mix and concentration of antibiotics within the patient for the correct amount of time. Now, this sometimes is in the hands of the patient, sometimes isn't. So, a case where it's in the hands of the patient, the doctor gives you an antibiotic regimen and says, okay, you need to take this every day at this time for this many days. Then the patient does really need to go through that whole entire regimen. Because if you just take it for a couple days and start feeling better and then stop taking it, what happens is, say it's a bacteriostatic antimicrobial, right? So it slows the pathogen down, but it doesn't kill them. So if you haven't taken the whole regimen, then you still have pathogens within your body, but the ones that are able to survive that regiment, then they're actually the stronger ones that are more likely to be antibiotic resistant. Same for bactericidal antimicrobials. You might take your antibiotics for a couple days and that will eliminate a number of the bacteria, but just the ones that are strong enough or have some resistance or remain. And if you don't take the regimen for the whole entire allotted period, then those ones will then be able to continue to survive and proliferate. Oftentimes there's a combination of antimicrobial agents so that there's broad coverage. And then we continue to develop new antibiotics, but we're kind of shooting ourselves in the foot if we are promoting antibiotic resistance as we're trying to develop new antibiotics. And so being really thoughtful of, as a system, how we're using antibiotics, and as patients, how we're using antibiotics, and then also how folks have access to antibiotics. So tuberculosis is a really interesting example. People have to take high levels of mixed antibiotics for long periods of times, and I'm talking about months, sometimes years. And so oftentimes patients are supervised in this so that they actually take their antibiotics. But if folks are not able to get to a clinic, or a clinician is not able to get to the patient, then that actually interrupts the regimen and leads to more likely antibiotic resistant tuberculosis strains, or tuberculosis causing agents. And so when we're thinking about this as patients, we're thinking about it also at a medical system and a societal level, really taking in all of those different variables and how we can decrease the trend towards more and more antibiotic resistant and multiple antibiotic resistant strains of pathogens. But we do have power, and folks in this class, many of you are going into healthcare settings. So understanding and utilizing this knowledge is really important. So we're going to go ahead and end chapter 10 there. Go ahead and review your notes, submit your discussion, which is just an overview of the mechanisms of actions and the various examples that we've used from our learning objectives, and submit that to D2L and see what other folks have learned from our discussion today. And I will see you in the next lecture. Okay, take care.